Welcome back to the deep dive. Today we're doing something a little different.
We are.
Usually we're looking at the macro world, economics, history, big tech, but today we're zooming in.
Way in.
Like literally, we are going microscopic.
We are. And honestly, this might be the most important shift in perspective we ever take. We're tackling the introduction to medical microbiology.
And before you check out, thinking this is just going to be some dusty list of Latin names and petri dishes, let me just stop you. I've been reading through the source material. We're using Murray's Medical Microbiology as our guide with some details from Javits. And the sheer scale of what we're talking about is, well, it's actually terrifying.
Terrifying is the right word.
Yeah.
But it's also, I think, awe-inspiring.
Let's start with the numbers, because they absolutely floored me. We walk around thinking we're the dominant species, right? The masters of our own little domain.
Yeah, that's the story we tell ourselves.
But according to Javits, there are an estimated 10 to the power of 30 microbial cells on Earth.
10 to the 30. That's a 1 followed by 30 zeros. It's a number so big, it's basically meaningless to the human brain. We just can't process it.
No. And get this. In the oceans alone, there are 100 million times as many bacteria as there are stars in the known universe.
Wow.
That just completely broke my brain. And the viruses. They're infecting and removing 20 to 40% of all of those bacterial cells every single day.
It's a microscopic Game of Thrones happening in every drop of seawater, constantly.
Exactly. It's a war zone.
It is a constant, silent, invisible war. And the crucial thing for you to realize is that this war isn't just happening out there in the ocean. It's happening in you right now.
That was the other stat that just, it got me. The bacteria in the average human gut weigh about 1 kilogram.
1 kilo.
That's like carrying around a bag of sugar.
Yeah.
But it's made entirely of living organisms inside your belly.
Exactly. And that sets the stage perfectly for what we're trying to do today. We're basing this whole deep dive on the foundational chapters from Murray's. And the mission here isn't just to memorize, bugs.
You can't be.
No. It's to understand that we are outnumbered.
We're the minority party in our own bodies.
In a way, yes. But your angle here, the host's angle, is that while all of this sounds like science fiction, it is the absolute bedrock of clinical medicine. You cannot practice medicine, you cannot understand human health without getting a handle on this invisible universe.
So if I'm the learner listening to this, a med student maybe, my goal isn't just to collect a bunch of facts, it's to learn the rules of engagement.
Correct. I want to shift your mindset. We're moving from memorizing lists of bacteria to understanding biological forces. Microbiology isn't just descriptive, it's a system of interactions. It is always about the invader, the microbe, versus the defender, the host. It's a chess match.
Murray uses this great metaphor for this. He talks about the clinician as Sherlock Holmes.
That is the core framework. That's the mental model you need. If you're a doctor, you are a detective. A patient comes in with a mystery, the disease. Your job is to profile the suspects. You need to know their habits, their fingerprints, their weaknesses, where they like to hide.
So today, we're building those profiles. We're going to look at the history of how we even found these things, which is a detective story in itself. And then we're going to break down the main suspects: viruses, bacteria, fungi, and parasites.
And we'll finish with the logic of diagnosis. How do you actually, catch the killer?
Okay, let's start with the history. And I know history can be dry, but Murray lays this out almost like a story. It's the story of us slowly realizing that the air around us isn't actually empty.
It really is a narrative. And for exams or just for, general literacy, you don't need every single date. But you need to know the milestones. Think of it in waves. First, you have the visualizers.
That's Leeuwenhoek, right, back in 1674.
Antony van Leeuwenhoek. And the amazing thing is he's a draper from Delft. He's not some university professor. He grinds his own lenses better than anyone else in the world.
Just a hobbyist, really.
A brilliant one. And he peers through this tiny, powerful lens at a drop of pond water and sees what he calls animalcules.
Little animals.
Exactly. Before him, this world literally did not exist to us. Can you imagine the psychological shift? Suddenly, water isn't just water, it's a zoo.
But, okay, seeing them is one thing. Organizing them is something else entirely. That brings us to the next wave, the organizers.
Right. Carolus Linnaeus. You can't study something if you can't name it. He gives us the system of genera and species we still use today. But here's the rub. Knowing they exist and giving them a name didn't mean we knew what they did. For a long, long time, people just thought these animalcules were there, like dust.
They didn't connect them to disease.
Not at all. The prevailing theory was miasma, bad air, or an imbalance of the four humors. The idea that a tiny, invisible animal could kill a grown man was, well, it was considered ludicrous.
So who finally proved it? Who are the provers?
This is the germ theory era. These are the heavy hitters. You have Friedrich Henle in 1840, who first proposed the criteria. He was the theorist. He basically said, look, if a bug causes a disease, you should be able to find it in the sick person. But he couldn't technically prove it.
Enter Robert Koch and Louis Pasteur in the 1870s and 80s.
The giants. Koch is he's crucial here. He developed what we now call Koch's postulates. This is the legal standard for convicting a germ.
Okay, walk us through that logic. It's so clean.
It is. So postulate 1, the organism must be found in all cases of the disease.
Makes sense.
2, you have to isolate it from the sick host and grow it in a pure culture. You have to get it by itself.
Okay.
3, you must put that pure culture into a healthy, susceptible host and cause the same disease. This is the big one.
And 4.
4, you have to re-isolate the same organism from that new sick host.
It's so rigorous. It's not just correlation. It's proving causation.
Exactly. It connects the suspect to the crime, definitively. They use this for anthrax, for rabies, for tuberculosis. Suddenly, TB wasn't a consumption of the spirit or a romantic ailment.
Right.
It was a bacterial infection, a specific enemy we could now see and name.
Okay, so we can see them, we can name them, we know they're guilty. Now comes the most important part for medicine. We need to kill them. This is the era of the executors.
The era of chemotherapy. And this is a huge memory anchor for you. Paul Ehrlich, 1910. He finds the first real antibacterial compound, salvarsan, to treat syphilis. He called it the magic bullet.
I love that phrase, the idea that you can shoot a bullet into the body that only hits the microbe but misses the human cell.
That is the holy grail of microbiology, selective toxicity. And then, of course, comes the most famous one, Fleming with penicillin in 1928.
Moldy Petri dish.
The mold that killed the staph on his plate. Serendipity, as they say, favors the prepared mind. Then you get Domack, who finds sulfanilamide in 35, and Waxman, who finds streptomycin in 43. And suddenly, we aren't helpless anymore. We have an arsenal. We have weapons.
There was one name in the virology section that stood out to me, though. John Enders in 1946. Why is he so important? I feel like he gets less press than Fleming.
He does, but he is arguably just as important for the 20th century. See, bacteria. Bacteria can grow in broth. It's basically soup. You put them in, they grow. Viruses are totally different.
They need living cells.
They need living cells. Before Enders, viruses were this black box. You couldn't study them easily. Enders was the one who figured out how to cultivate viruses in cell cultures, basically, keeping human cells alive in a dish so the virus had a home to grow in.
And without that discovery?
Without that, you don't get the polio vaccine. You don't get the measles vaccine. You don't get modern virology as we know it. That single technical leap paved the way for saving millions, maybe billions of lives.
And now, where are we now? The modern era.
Which is the genomic era. We've moved from just looking at things through a microscope to being able to read their entire genetic code. It's faster, it's cheaper, and it's revealing this huge unseen microbiome. All those bugs we can never grow in a lab because, I don't know, they die when exposed to oxygen, right, or they need some weird nutrient we don't understand.
But now we know they're there.
We know they're there because we can see their DNA signatures in the sample.
So we've gone from there's nothing there to there are billions of things there, and we can read their source code.
Precisely. It's a whole new world.
Okay, so that's the timeline. Now let's meet the suspects. We're going to start with the smallest and honestly the weirdest of the bunch, the viruses.
Right. Section 1, the viral spectrum. And Mary calls them true parasites.
But calling them parasites almost feels too generous, doesn't it? A parasite is usually alive. These things are closer to, I don't know, zombie code. Murray describes them as inert information.
That's a great way to put it.
They aren't even alive until they hijack us.
That's the key philosophical distinction. A bacterium can often live on a table or in the soil. It is its own metabolism. A virus has zero life without a host cell. It is a set of blueprints waiting for a construction crew. If it doesn't find a cell to enter, it's just dust.
And they're unbelievably small, 18 to 600 nanometers.
Yeah, too small for a standard light microscope. That's why Leeuwenhoek never saw them. You need an electron microscope to even get a picture.
Let's talk about their structure because this really dictates how we fight them. I always just think of them as a little shell with some stuff inside.
That's basically it. They have a payload and they have armor. The payload is their nucleic acid. It's either DNA or RNA. And as a general rule for Murray, it's one or the other, not both.
Right.
This is their soul, their genetic instructions.
And the armor.
It's a protein shell called a capsid. This protects the genetic material. Now, here is a crucial distinction that comes up in exams and clinical practice all the time. Some viruses are naked, just the capsid and the payload, and some are enveloped.
Okay, and the enveloped ones, they wrap themselves in a lipid membrane that they steal from our own cells, right? It's like wearing the skin of your victim as a disguise.
That is a terrifying image, but it's completely accurate. But here's the clinical plot twist. That envelope, that disguise, it actually makes them weaker, not stronger.
Wait, really? I would have thought an extra layer makes you tougher.
It's totally counterintuitive. But think about what a lipid envelope is. It's a fatty, oily layer. What happens to oil when you use soap or alcohol-based hand sanitizer?
It dissolves, it breaks apart.
Exactly, or if it just dries out. So enveloped viruses like influenza, like HIV, like COVID-19 are actually very fragile in the environment. They need to stay wet, and they are easily killed by hand sanitizer.
Whereas the naked viruses.
Like norovirus, the
Yeah.
cruise ship virus. They're just a tough protein shell. Alcohol doesn't hurt them very much. Drying out doesn't bother them. That's why norovirus can rip through a cruise ship so easily. It survives on a door handle for days.
That is a massive aha moment for me. The structure dictates its survival strategy.
Precisely. And because viruses are so simple, the clinical disease they cause depends entirely on the target tissue. Is this virus programmed to target your nose? You get a cold. Is it targeting your liver cells? You get hepatitis. Is it targeting your CD4 T cells? You get HIV. They're like guided missiles.
Okay, but as I was reading the Jowett supplement, it seems biology never likes simple rules. There are rule breakers.
Never. And these rule breakers are high yield for differentiation questions on exams. They love asking about the exceptions.
First up, giant viruses like mimivirus and megavirus.
These things are huge, 400 to 500 nanometers. You can actually see them with a light microscope, which breaks that size rule we just talked about. And their genomes are massive for a virus. They encode things like metabolic enzymes, which viruses are not supposed to have.
So they blur the line between a virus and a simple bacterium?
They do. It even suggests that maybe viruses de-evolved from more complex cellular ancestors. It's a fascinating idea.
Then you have viroids.
These are mostly plant pathogens, but they're important conceptually. Think of them as naked RNA. There's no protein coat, no capsid. It's just a single circular loop of infectious RNA that gets into a plant cell and completely messes up its machinery.
But the one that really it keeps me up at night is prions, the zombie protein.
Prions are one of the most fascinating and terrifying concepts in all of biology.
Yeah.
This is a protein-only infectious agent. No DNA, no RNA, just protein.
How is that even possible? I thought the central dogma of biology was DNA makes RNA makes protein. How can a protein replicate itself without any genetic instructions?
That was the dogma. Prions broke it. Here is the mechanism, and you need to lock this in because it's a classic exam concept. So in your brain, you have a normal healthy protein called PRPC. It has a shape that's mostly alpha helical. Think of it like a slinky. It's flexible, it's soluble, it's doing its normal job.
Okay, PRPC, alpha helix, the good guy.
Then you somehow introduce the infectious version, the isoform, called PRPSC. The SAC is for scrapie, a prion disease in sheep. This version has the exact same amino acid sequence, but it is folded differently. It's rich in beta sheets.
Beta sheets, so it's flat, rigid, that's the bad guy.
And here's the zombie part. When the bad PRPSC touches a good, normal PRPC, it physically forces it to flip its shape. It converts the good protein into another bad protein. It's like a zombie biting a human. Now you have two zombies, then they go and bite two more. It's a chain reaction.
And why is that beta sheet shape so bad for the cell?
Because of thermodynamics. These flat beta sheet proteins are incredibly stable, and they love to stick together. They stack up like Lego bricks, forming these insoluble clumps called amyloid plaques. Our body's enzymes, our proteases, are designed to chew up old alpha helices. They can't bite through these beta sheet stacks. They're indestructible garbage that just builds up and up inside the neuron.
So the brain cell fills up with this indestructible trash and eventually dies.
Exactly. And it leaves a hole behind. That's why we call these diseases spongiform encephalopathies. Sponge brain. You see it in Kuru, in Creutzfeldt-Jakob disease, or CJD, and in mad cow disease.
And the truly scary part is that you can't kill them easily.
Right. Because they have no DNA, UV light doesn't work. Because they're so tightly folded into these beta sheets, standard heat sterilization often fails. Auto-cleaving surgical instruments sometimes isn't even enough. They are the ultimate survivors.
Okay, I'm shaking off the prion horror. Let's move up the size chart to the prokaryotes. Let's talk about bacteria.
Section 2, the prokaryotic world. This is the bread and butter of clinical microbiology.
And the biggest distinction here is prokaryote versus eukaryote. And this isn't just trivia, it's the reason we can take antibiotics without, killing ourselves.
That's it, exactly. Prokaryote means before nucleus. They have no nuclear membrane, they have no mitochondria, no Golgi apparatus, no endoplasmic reticulum. They are just streamlined survival machines.
And because they are so fundamentally different from our cells, which are eukaryotes, we can target those differences.
Absolutely. For example, their ribosomes, the little factories that make protein, are a different size and shape in ours. So we can build drugs like tetracycline that are shaped to jam their ribosomes but don't fit into ours. That's selective toxicity again.
But the single most important structure for a doctor to know is the cell wall. This is the big Gram-Stain divider.
If you learn nothing else about bacteria today, learn this. The Gram-Stain divides almost all bacteria into two massive camps based on their armor. This single test dictates everything from initial diagnosis to treatment.
Okay, so camp 1, gram-positive.
Think positive equals purple equals peptidoglycan. Peptidoglycan is this mesh-like layer made of sugars and amino acids. In gram-positive bacteria, this layer is incredibly thick.
And what's happening mechanically during the stain? Why does it stay purple?
Think of that thick peptidoglycan layer, like a thick wool sweater. You flood the cell with crystal violet, which is a purple dye, and then you add iodine. These two chemicals link up inside the cell to form these giant crystals. In a gram-positive cell, that thick wool sweater physically traps those massive crystals. They literally cannot wash out. It's a physical entrapment.
Okay, so that's why it stays purple. What about camp 2, gram-negative?
These guys have a very thin peptidoglycan layer. The sweater is threadbare. So when you do the decolorizing step with alcohol, those big crystals just fall right out. They lose the purple color. Then you add the pink counterstain, saffronin, and they pick that up.
But the gram-negatives have something else, right? A kind of secret weapon.
They do. They have an outer membrane. It's a second lipid bilayer, a second skin outside that thin cell wall.
And why does that matter clinically?
Two big reasons. First, that outer membrane is a shield. It can block certain drugs like vancomycin, which is a very large antibiotic, from ever reaching their target. It acts as a barrier. Second, that outer membrane contains something called endotoxin, or LPS, which is a potent trigger for septic shock. So, as a general rule, gram-negative infections can be harder to treat and more dangerous when they get into the bloodstream.
Are there exceptions to this whole gram-stain thing?
Always. There's Mycobacterium, which causes TB. It has this waxy, complex wall that doesn't stain well with a gram stain. You need a special acid-fast stain for them. But the big exception that Jaywitz highlights is Mycoplasma.
Mycoplasma is the nudist of the bacterial world.
Essentially, yes. It has no cell wall at all. It never evolved one. So think about it. An antibiotic like penicillin works by attacking the cell wall. If you try to treat a Mycoplasma pneumonia with penicillin, what happens?
Nothing. It's like trying to break a window in a house that has no windows.
Precisely. And that is a classic exam question. Mycoplasma is intrinsically resistant to all cell wall active agents. It also does something else weird. It steals cholesterol from the host cell membranes to help stabilize its own membrane, which other bacteria generally don't do.
Now, Murray brings up this concept of the microbiome again here. He calls it an organ.
Which is a huge paradigm shift. We used to think of our gut bacteria as just, hitchhikers or commensals, which means eating at the same table. Now we realize they function collectively, like a liver or a kidney.
How so? What do they do?
They metabolize drugs we take. They produce essential vitamins like vitamin K. They digest complex carbohydrates that our own enzymes can't touch, and they turn them into short-chain fatty acids that actually feed our colon cells. They even communicate with our brain, the gut-brain axis.
So when we take a course of broad-spectrum antibiotics, we're essentially, causing organ damage?
Yes. You're bombing a rainforest to kill one snake. And when that ecosystem is damaged, opportunistic weeds can take over. That's what C. difficile is.
And when that organ gets sick, we can treat it with a transplant.
Exactly. That's what a fecal microbiota transplant, or FMT, is. We usually use it for recurrent C. diff infections right now, but researchers are looking at it for treating IBD, metabolic syndromes, maybe even obesity. You're literally transplanting a healthy ecosystem into a damaged one.
Speaking of ecosystems, there was a concept in Joette's that I just loved. Bacterial communication. I always pictured bacteria as these lonely, isolated cells.
Far from it. This is quorum sensing. And it's a very high-yield concept for exams. Bacteria are not just swimming around randomly. They are constantly chatting with each other.
How do they talk? Do they send texts?
Chemical texts. The most common one mentioned in the books is AHL acylated homoserine lactone.
AHL. Okay, how does that work?
Okay, imagine you have bacteria in a test tube. They're all constantly secreting small amounts of this AHL molecule, just whispering, I'm here, I'm here. When there are only a few of them, the concentration of AHL in the fluid is very low. The signal just drifts away.
But when there's a crowd?
When the population density hits a certain threshold, a quorum, the AHL concentration spikes. It gets high enough to diffuse back into the bacteria and bind to transcription factors. This flips a genetic switch in all of them at the same time.
It's like a flash mob. They're all waiting for the signal to start the dance.
That's a perfect analogy. They suddenly switch on coordinated group behaviors.
Yeah.
They might all decide to form a biofilm, which is like a slime city that protects them from antibiotics. Or they might all decide to release their toxins at once. They wait until they have the numbers to overwhelm the host defenses before they reveal their attack. It's tactical.
That is just wildly sophisticated for a single-celled organism. It implies a level of strategy we don't usually credit them with. Okay, let's level up again. Eukaryotes. Now we have a nucleus. We're talking fungi and protists.
Section 3. The eukaryotic shift.
Yeah.
And the key thing to remember about fungi is that they are much more like us than bacteria are. They have mitochondria, a Golgi apparatus, the works. And this makes them harder to treat. Because if you're trying to find a drug to poison their cellular machinery, you run a much higher risk of poisoning your own. The targets are more similar. There's less selective toxicity.
But they are shapeshifters. Murray talks about this concept of dimorphism.
This is exam gold. You will see this. Fungi can exist in two main forms. Yeast, which is a single cell, or mold, which is filamentous and fuzzy.
Right. Yeast is unicellular. Mold is made of filaments called hyphae.
Correct. And for the really dangerous systemic pathogens, the ones that can get inside you and kill you, the form they take depends on the temperature.
What's the rule for that?
The mnemonic is mold in the cold, yeast in the beast.
I love that. Unpack it for me.
In the environment, the cold, say 25 degrees Celsius in the soil, they grow as a mold with hyphae. This form is good for spreading spores in the wind, but when you inhale one of those spores and it gets into your lungs at body temperature, the beast, 37 degrees Celsius, it transforms into a yeast sphere.
Why does it switch?
It's a survival mechanism. The yeast form is much better at traveling through the bloodstream and evading the immune system. It's like an adaptive camouflage. Key examples are histoplasma, blastomyces, coccidioids. If you see an exam question describing something that grows as a mold on a petri dish, but is seen as a yeast in a lung biopsy, the answer is thermal dimorphism.
Got it. Now, let's add some protists to this eukaryotic mix, specifically algae. I usually think of algae as pond scum, not a medical problem.
And usually they aren't. They don't typically infect us directly, but you need to watch out for the toxins they produce, specifically from red tides.
Ah, these are the dinoflagellates.
Exactly. They can bloom in the ocean and produce these incredibly potent neurotoxins like saxitoxin.
Saxitoxin is bad news.
Very bad news. It blocks sodium channels. And remember, your nerves need those sodium channels to fire action potentials. If you block them, you get paralysis. So the shellfish eat the algae, they concentrate the toxin in their tissues, and then you eat the shellfish.
And you get paralytic shellfish poisoning.
Right. Numbness, tingling, ataxia, and in severe cases, respiratory failure. It's not an infection, it's an intoxication. You aren't fighting a bug, you're fighting a poison.
But there was one algae that actually does infect people, wasn't there?
A very rare one, yes. Prototheca. It's a strange one. It's an algae that lost its chlorophyll. It basically gave up on photosynthesis and decided to become a parasite instead. It can cause skin infections or bursitis, mostly in dogs or cattle, but sometimes in immunocompromised humans. It's the rare exception to the algae don't infect rule.
All right, moving on to the real heavyweights of this group, parasites.
Section 4, the parasitic spectrum.
The size difference here is just comical. We go from a single-celled protozoa that's like 4 microns to a tapeworm that can be 10 meters long.
Imagine a microbe that is 30 feet long living inside your intestine. That is the incredible scope of parasitology.
And their life cycles sound like something out of a Rube Goldberg machine.
They are by far the most complex. A parasite might need to live in a snail, then a fish, then a human to complete its life cycle. Take schistosoma. It lives in a specific type of snail. It releases larvae into the water. Those larvae then drill directly into your skin while you're swimming. They migrate to your liver. They mature, find a mate, and then move to your bladder or your intestine to lay thousands of eggs.
It's exhausting just hearing about it. Why go to all that trouble?
Evolution. It allows them to exploit different environmental niches, but it also creates vulnerabilities for them. If you can eliminate the right snail from the water supply, you can stop the disease.
Which brings us to diagnosis. For bacteria, we do a culture. For viruses, we might run a PCR, but for parasites.
History is king, specifically travel and diet, because parasites are often very regionally specific. If you have a patient with fever and liver problems who just came back from swimming in the Nile River in Egypt, you have to be thinking about schistosomes. If they've never left Ohio, you aren't. You have to ask where they've been and what they ate.
So we've met all the suspects: viruses, bacteria, fungi, parasites. Now let's look at the battlefield itself. Section 5. Immunology. The host response.
And Murray calls the immune response a double-edged sword. This is such a critical concept. We tend to think immune system equals good, infection equals bad.
But that's too simple.
Way too simple.
Yeah.
Because also the symptoms you actually feel, the fever, the swelling, the pus, even the tissue damage, are caused by your own army, not by the invader. The immune response itself causes the pathology.
It's friendly fire.
It's collateral damage. When you get the flu, the virus isn't what's making you ache all over. Your body's massive release of chemicals like interferon is what's making you ache. It's the cost of waging war.
And that war has two main fronts: innate and adaptive immunity.
Innate is your first responders. It's ancient. It's your skin barrier, your stomach acid. Then you have the cells, neutrophils and macrophages. They're programmed to recognize broad patterns. They see something and think, this looks like a bacteria, kill it. It's very fast, but it's nonspecific. It's a shotgun blast.
And adaptive is the sniper.
Adaptive is the special forces. This is your antibodies and your T cells. It takes time to spin up days or even weeks because it has to engineer a specific weapon for a specific enemy. But once it does, it's incredibly precise. And it remembers. It has memory.
Murray also makes a key distinction about how the body fights differently depending on where the bug is hiding.
Right. This is the tactical decision point. If the bug is extracellular, so it's outside your cells, like staph swimming around in your blood, you need to eat it. You send in the phagocytes, the eater cells, and you use antibodies to tag the bacteria for destruction.
And if it's intracellular, like a virus or maybe chlamydia that's hiding inside one of my cells?
Then the eaters can't reach them. The bug is hiding in the bunker. You need a different strategy. You need your NK cells, your natural killer cells, and your cytotoxic T cells. Their job is to patrol your body, scan your cells, find the infected ones, and then give them the command to commit suicide. Apoptosis. You have to burn down the factory to stop the virus production.
That's brutal. It's a scorched earth policy.
It is. And vaccines. Vaccines are essentially intelligence briefings for your immune system. You show the immune system a mugshot of the enemy, an antigen, so that when the real war starts, your adaptive response is already mobilized. You skip that slow initial phase. You have the snipers in position before the invasion even starts.
Okay, so we have the suspects, we have the defense. Now, put us in the white coat. Let's talk about Section 6. Diagnostic microbiology and clinical logic.
This is the Sherlock Holmes framework that Murray lays out.
Yeah.
4 specific questions that every doctor must ask in order.
Let's apply this to a case. A patient walks into the ER. They have a fever, a stiff neck, a bad headache.
Okay, classic presentation for meningitis.
Question 1. Is it an infection?
Sounds obvious, but it's not. Could this be a drug reaction? Could it be a brain bleed? Could it be some autoimmune disease? You have to look for the signs of inflammation. A high white blood cell count, a fever, high C-reactive protein. You have to prove that a bug is likely involved.
Okay. Question 2. Where is the infection?
This determines your sample. If you suspect meningitis, an infection around the brain, a urine sample is useless. You need to get spinal fluid. You have to go to the scene of the crime. If you suspected pneumonia, you'd want sputum from the lungs.
Question 3. Which microbe is it?
This is the identification step. You take that spinal fluid to the lab, you do a gram stain on it. Do you see little purple spheres in chains? Maybe strep pneumo? Do you see pink kidney bean shapes? Maybe Neisseria? Or maybe you see nothing on the gram stain, and now you need to run a PCR to look for viruses.
Murray adds a really important nuance here. The shift from what is it to how is it causing disease?
Right. Is it releasing a toxin? Is it directly invading and destroying tissue? This helps you understand the prognosis and guide treatment.
And finally, the big one, question 4, should it be treated?
And if so, how? This is a trap for young doctors. Just because you find a bacteria doesn't automatically mean you treat it.
Why not? If I find a bug, shouldn't I kill it?
Not if it's just colonization. We all have Staphylococcus living harmlessly in our skin. If you swab a healthy arm and find staph, you don't give that person antibiotics. You only treat if it's causing disease. And of course, if it's a virus, most of the time antibiotics won't do anything at all.
And throwing a big, broad-spectrum antibiotic at everything is bad practice.
It's the core of antimicrobial stewardship. If you use a nuke when you really needed a scalpel, you destroy the patient's microbiome, that good organ we talked about, and you help breed antibiotic resistance. Specificity is always key.
One last pattern recognition tip from Murray. Thinking about infections as either exogenous or endogenous.
That's a great way to categorize disease. Exogenous means it came from the outside. The flu virus, tetanus from a rusty nail, gonorrhea, you catch from the world.
And endogenous.
It comes from inside you. It's your own normal flora ending up in the wrong place. E. coli belongs in your bowel. It's a good citizen there. But if it gets into your bladder, you have UTI. If it gets into your abdominal cavity from a burst appendix, you have peritonitis.
And Murray says that most human bacterial diseases are actually endogenous.
That's right. We are most often attacked by our own passengers. The line between friend and foe is just a matter of geography inside the body.
That is a very sobering thought to end on. So, let's wrap this up. We've gone from the invisible animalcules of Leeuwenhoek all the way to the genomic revolution. We've looked at the whole spectrum from tiny viruses and zombie pions up to massive 10-meter tapeworms.
And the key takeaway from all of it, we are not isolated individuals. We are walking ecosystems.
That's the final provocative thought for all of you listening. If our own human cells are outnumbered by microbial cells, and if those microbes influence our digestion, our immunity, and maybe even our mood, are we truly individuals, or are we just the host vessel for a vast microbial civilization?
The future of medicine might not be about killing bugs. It might be about farming them, cultivating the right ecosystem to keep the whole vessel healthy. Farming, not fighting.
I love that. Okay, listeners, don't go anywhere. We need to lock all of this in. It is time for the recall quiz.
We have 6 questions. I'll read the scenario. We'll pause for a moment for you to think, and then I'll give you the answer and the reference. These are all single best answer style.
Let's do it.
Question 1. A degenerative neurological disease is attributed to a prion. What is the fundamental molecular change responsible for the pathogenicity of this agent? Pause. The answer is the conversion of an alpha-helical host protein, which is PRPC, to a beta-sheet-rich infectious form, PRPSC. Remember, this conformational flip is what creates the protease-resistant amyloid plaques. You can review this in Jowett's Chapter 1 in the section on prions.
Beta sheets are bad. They stack up and can't be broken down. Got it. Next one.
Question 2. A patient presents with a systemic fungal infection. The organism grows as a mold at 25 degrees Celsius, but it's a yeast at 37 degrees Celsius. Which term best describes this property? Pause. The answer is dimorphism. Remember the mnemonic, mold in the cold, yeast in the beast. It switches its form to survive inside the host. That's in Murray Chapter 1, the section on fungi.
Okay, question 3.
Question 3. In the formation of a biofilm, bacteria monitor their own population density to regulate gene expression. What is the name of the diffusible signal molecule used in this process? Pause. The answer is acylated homoserine lactone, or AHL. This is the quorum sensing signal. It accumulates until it gets to a high enough concentration to trigger that group behavior. You can find that in Jowett's Chapter 1 in the section on prokaryotic communities.
The flash mob trigger. Check.
Question 4. A patient presents with paresthesias, that's a tingling sensation, and ataxia after eating shellfish. The symptoms are caused by a neurotoxin accumulated by the shellfish. What is the causative eukaryotic microorganism? Pause. The answer is dinoflagellates. They produce toxins like saxitoxin during red tides, and these toxins block sodium channels in your nerves. Reference for that is Jowett's Chapter 1, section algae.
2 more to go.
Question 5. Who was the first scientist to successfully cultivate viruses in cell cultures? A discovery that facilitated large-scale vaccine production. Pause. The answer is John Enders back in 1946. Remember, before him, we couldn't grow them effectively in the lab. He was the one who unlocked the door to modern vaccines. That's in Murray Chapter 1 in the historical perspective section.
And the last one.
Question 6. Which bacterial structure is the primary differentiator between gram-positive and gram-negative organisms and is notably absent in the genus Mycoplasma? Pause. The answer is the cell wall, which is made of peptidoglycan. Remember, Mycoplasma has no wall at all. Gram-positives have a thick wall that traps the purple dye, and gram-negatives have a thin wall plus an outer membrane, so they lose the purple dye. And you can review all of that in Murray Chapter 1 in the section on bacteria.
And there you have it. You have just survived the introduction to the invisible universe.
This is just the survey. In the next deep dive, we start cracking the cases one by one.
Can't wait. We'll see you then.